Welcome to the third lecture in this 2009 series, Global Diseases, Voices from the Vanguard. This is a joint effort between the Center for Tropical and Emerging Global Diseases and the Knight Chair in Health and Medical Journalism, Pat Thomas of the Grady College of Journalism and Mass Communication. And I'm pleased that each of you are here. I know outside it's very tempting, the first nice day in about 4 days. As most of you know, the lecture series is intended to create interest in global diseases across the campus, from north to south campus. And today we have Dr. Barney Graham who will talk to us. But let me first remind you that we have a reception immediately following the lecture over in Demosthenian Hall right next door, and that the next Voices from the Vanguard will be April 14th, and that'll be Dr. Jennifer Friedman from Brown University, and she will probably talk to us about her work in the Philippines on a variety of neglected tropical diseases. So tonight, Pat and I have asked Dr. Barbara Schuster to introduce Dr. Graham to you. But before I do that, it's first a distinct pleasure to introduce Dr. Schuster to you. Dr. Barbara Schuster is the new campus dean of the Medical College of Georgia, University of Georgia Medical Partnership campus in Athens. She's a proven educator, physician, and administrator. Barbara comes to us from Wright State University in Ohio, and there she chaired internal medicine and has most recently taken a year to work with the American Medical Association in terms of doing some research. So it's a pleasure to introduce her. She's new to the campus, and I think it's terrific that she took the time out from putting together a medical school, which is literally what she's doing, to be with us tonight. So it's my pleasure to introduce Barbara to you. I'd ask you to welcome her and her husband, Richard, who is a professor of UGA's College of Public Health. And they're both here tonight, so please introduce yourself to them, too. Thank you, Barbara.
Thank you very much for that wonderful introduction. My husband is here in the second row with me, so I hope I'll get to meet each of you later after Dr. Graham presents. It is really my pleasure to introduce a colleague. I don't know Dr. Graham, though we have mutual friends in Rochester, New York, people who also work in the field of virology. But Dr. Graham is a graduate of Kansas, the University of Kansas, where he received his MD degree after graduating from Rice University. He went on to do post-medical school work called residency at Vanderbilt and completed a fellowship there after internal medicine in infectious disease. The infectious disease folks are always close to my heart because had I chosen a specialty, I had thought about infectious disease. And so microbiology and infectious disease is something I've always had an interest in. He, of course, has, rose in the ranks, became a professor, and has done outstanding work in the fields of RSV, also known as respiratory syncytial virus, and HIV. And today he's going to speak to us on the HIV vaccine development. What is the next step? So please welcome Dr. Graham.
Well, thank you very much, and it's my pleasure to be here. I need to tell you a little bit about my connection to Dr. Colley because he rescued me during my dissertation work when I was getting my PhD at Vanderbilt. And even though I was a virologist and wanted to be a virologist, and he was a parasitologist, he helped me finish that dissertation project. And I think it may be his only virology paper ever, but he does have one. The thing I learned there, though, was that most of the really big ideas about immunology have come out of the parasitology field. And it's mostly things borrowed from parasitology that has populated the viral immunology literature, and so I owe him a debt for that as well. Okay. And I thought of him not too long ago when I was in Uganda and had an opportunity to go rafting on the Nile River. And they said it was a Class 5 rapids, and so the schisto really shouldn't be there because they should only be present in very calm waters. And so we did go rafting. I was at the head of the boat, and we had a terrific time, and my antibody titer a few months later was negative. But I did think of you during that trip. So why do we need an HIV vaccine? This is the main reason, that there's still more than 7,000 infections every day occurring throughout the world. And despite the delivery of antiretrovirals in a more efficient way globally, there's still about 10 new infections for every new person that started on antiretrovirals. And if that is the trend, we're never going to be able to treat our way out of the HIV epidemic. Unfortunately, many of these infections are in low and middle income countries, and they are in our most vulnerable populations, women and young children. In the United States, the epidemic has not gotten as much attention recently as I think it should have, but it still is significant. There's still 55 to 65,000 new infections every year. And as you see, it's moved more into minority populations, more vulnerable populations, and it's become a disease of young people, particularly within those minority populations. So we do need a vaccine. So why do we think vaccines would work for HIV? The main reason is because of their track record. Vaccines have been our most cost-effective and efficient way of managing and controlling infectious disease epidemics, and I'm showing you just a few examples here of vaccines for viruses. They're the year of the peak incidence of that infection in the United States, the number of cases present during those years. For instance, mumps in 1967, almost 200,000 cases. In the early 60s, 500,000 cases of measles. Those diseases have almost vanished and have become very rare, except for last year when there were 7,000 cases of mumps because people stopped taking their vaccines because they forgot how bad mumps could be. We've eradicated smallpox. That happened in 1980, so no place on earth is having smallpox now, and polio is very rare, if ever present, in the Western Hemisphere. So vaccines do work. Vaccines work even when they're partially effective or partially administered, and so this is an example from the mumps epidemic. When the vaccine became available in 1967, before very many people were immunized, the epidemic started going away. And it's one of our best examples of how just taking a few transmitters and a few susceptible people out of the population, these kinds of epidemics that are requiring a large number of susceptible people for transmission can start going away. So even before the recommendation was made for some people to be vaccinated in a more universal way, the epidemic was vanishing. But there are problems for HIV, very significant biological hurdles that have not been accomplished in any other vaccine effort. And so HIV has a list of problems. For instance, the lack of natural immunity. There is not an example of where our natural immune response was able to fully clear virus from an infected human being. And so the absence of the ability to study the natural clearance mechanisms is not present in HIV. There's also evidence that if you're infected with one strain of HIV, you can be infected with another strain of HIV. So natural virus infection doesn't necessarily prevent you from having another natural virus infection. The genetic diversity is difficult for two major reasons. One is the question of what do you put in the vaccine? How do you select the antigens for the vaccine when there's so many tens of thousands of choices to make? And secondly, because if you do become infected, even though you're vaccinated, the genetic variation in the virus growing in you can help you escape those immune responses. So it's problematic on both the population and the individual level. Thirdly, there is an infection and a deficit created in the first responders for the immune system. Both antigen-presenting cells are impacted, and the CD4 T cell that organizes the rest of the immune response are either destroyed or their function is diminished. And then HIV can infect immunoprivileged sites like the eye, where the immune system has a hard time reaching. It can become integrated into your genome and become latent, and so it can exist in a state in which no antigens are seen so that the immune response can't see it. Or it can become sequestered. So a lot of HIV in an infected person is actually sitting outside of your cells. It's sitting attached to the outside of dendritic cells and the lymph nodes, and that's not a place where it can be cleared by the immune system. And then for vaccine developers, one of the biggest problems for HIV is the failure to induce neutralizing antibodies, which is the basis for most effective vaccines that we have today. So I'll go through why despite these biological problems, I think it's still possible to have an HIV vaccine. And then I'll tell you about some of the difficulties we've had in the HIV vaccine development field the last 2 years. And then at the end, I'll let you help me decide what we should do next. What should be the next step? So when you're infected with the virus, it's interesting that despite the diversity of viruses that a person might be exposed to, the typical infection is a single virion crossing over and infecting the next person. We know that because sequencing virus early in that infection period only gives you a single genotype. And it takes about 4 to 6 weeks before the genotypic diversity to start developing. And so unless a person has ulcer disease, most infections occur from just a single virion. And so the transmission efficiency is not that high with HIV. It's not a very contagious virus, and it may be that a very minimal immune response of the right type in the right place could have a big impact on HIV transmission. So when it infects usually through a mucosal surface. A dendritic cell grabs the virus and feeds it to a CD4 T cell nearby. And within just a few hours, it is spread and can be detected in regional lymph nodes. And within a few days, it is systemically spread, and you can find it in all the lymph nodes of the body. And at that time, it achieves a dynamic equilibrium where there's a lot of virus being made and a lot of virus being cleared, but you reach a set point where you have a steady state virus level. But that process of making virus and killing virus is what eventually erodes and destroys your lymph node architecture and develops and allows you to develop immunodeficiency. We know that if we had an antibody that was effective at blocking HIV, that could neutralize HIV, we could potentially block infection. And that would be our best option. That's how most good viral vaccines work. But as I'll explain later in more detail, we do not have antigens identified that can elicit broadly neutralizing antibodies to HIV. So antibodies that can block the most commonly transmitted strains of HIV are difficult to identify. We also know that when we have our normal adaptive immune response, particularly the T cell response, when it happens, it cannot fully clear virus. It just achieves this steady state of virus persistence. And we know that in the meantime, before that T cell response can occur, this state of latency can be established and sequestration. So the big question for vaccines, as we're now working on vaccines, is what happens if you move the T cell response sooner? If the adaptive immune response can happen a little bit earlier and a little bit higher magnitude, what effect could that have on virus replication? So when you're infected with HIV, it takes about 3 to 4 weeks for the virus to reach its peak point of viremia. And at that point, the T cell response kicks in, and a specialized type of T cell, the CD8 T cell, the killer T cell, or the CTL, cytotoxic T lymphocyte, is able to recognize the virus and start clearing virus-infected cells, and that's what reduces the viral load to this set point of viremia that you would have for 7 to 10 years untreated before you developed HIV or before you developed AIDS. If we had an antibody response, preformed antibody response, we might be able to block infection. But it's not likely that we'll be able to generate an antibody response like that in the near future. So most of the vaccines that were being tested right now are T-cell-based vaccines, hoping that these specialized CD8 T cells that recognize processed virus in the surface proteins called MHC molecules, type 1, class 1 MHC molecules, can rapidly clear those virus-infected cells, and then, if potent enough, could potentially clear your body and have an abortive infection, but more likely, as what's seen in animal models, reduce the peak viremia and achieve a lower set point viral load that results in a very delayed illness progression. You might also have a situation where it doesn't really reduce viral load in the host, but maybe it reduces the output virus and maybe reduce transmission to the next person. That could also have a beneficial effect on the epidemic. And then, because things don't always go the way you want them to go, there's also the possibility of disease enhancement. That's been something that's happened with respiratory syncytial virus in the past, with measles virus in the past, with some coronaviruses in the past, and I'll tell you a story about HIV pretty soon. So when you think about starting to make an HIV vaccine, and you look back at history, how long does it take to make a vaccine? Can you do it even in one career? And the answer is only if you're lucky and you start soon, start early, because it usually takes decades to develop a vaccine. From the time a virus is discovered to the time those vaccines are available for human use is usually measured in decades. And right now we're at 25 years and counting for HIV, but there's other difficult viruses with difficult biology that we also still haven't conquered, and they're common viruses, like RSV is more than 50 years and herpes simplex virus is more than 90 years. These are viruses that have properties of latency, like herpes, or that inactivate some of the early immune responses that, like RSV, that share these properties with HIV. So not all viruses are easy. In 1997, President Clinton gave an address at Morgan State commencement and said, maybe we should put more effort behind the HIV vaccine development process, and proposed that a new center be built on the NIH campus. And so that project was started in '98. The building was finished in August of 2000, and we started occupying these laboratories. I went from Vanderbilt University at that time to help start this center, and partly because it has unique features. Most of the easy vaccines have already been made for the viruses that we have, and the formula, the risk-benefit and the cost-benefit formula now for vaccines is such that it may not be that industry alone will be able to develop very many more vaccines. And so the unique feature of this, of our program, is that there is government money, public money for public health in vaccine manufacturing. That has not occurred very often. If it has occurred, it's mostly been in the setting of the military. It's a very mission-oriented program, unlike many programs at NIH that are more free-flowing basic research, follow the idea. This is a very mission-oriented group. And self-contained means that we cover a broad array of activities, all the way from atomic structure of the proteins to clinical trials in a relatively small group. So these are some of the principal investigators at the VRC. We work out of Building 40 on the NIH campus. But now the program also includes what we call the pilot plant, which is a GMP production facility in Frederick, about 20 miles north of campus. It's a state-of-the-art facility that's every bit like you'd see at a Merck or a Glaxo. There's a clinic here now in this building. This is the new Building 10, so we can do phase 1 clinical trials. This is the clinical trials group. And we also have a processing site in Gaithersburg, a few miles away, that processes our clinical samples and does good laboratory practice endpoint analysis. We also can do small animal work, and we have a primate colony spread out throughout the world of about 500 Indian rhesus macaques that allow us to do the studies we need to do preclinically before we get into clinical trials. And because the gates are imposing and the fence around NIH is imposing, we now have a mobile clinic to see if we can reach out beyond the gates to do these clinical trials. And since 9/11 in 2001, our mission has expanded somewhat. And I won't say anything about these other programs, but in addition to HIV, we now have programs or have done programs on Ebola, Marburg, smallpox vaccine, alternative smallpox vaccines, West Nile virus, SARS coronavirus, and influenza viruses. So the story of HIV vaccine development has been kind of staggered, and it has its ups and downs. Pat Thomas has written a book about some of these ups and downs, some of the peaks and valleys that she's witnessed, but there's been a clinical trials program for HIV vaccine evaluation since 1987. Vanderbilt was one of the founding sites. And since that time, a number of different vaccine products have been evaluated, maybe as many as 60 different concepts. But those have only resulted in a small number of efficacy trials where you actually test to see whether a vaccine works or not. And the first one was this VaxGen GP120, which was based on a purified protein. It was based on the same thing that the hepatitis B vaccine was based on. And the problem is, because we weren't inducing antibodies that could neutralize the common virus, it did not work. And so that trial ended in 2003. A purified protein can induce antibody and CD4 T cells, but it cannot induce CD8 T cells, the ones that can clear these virus-infected cells. And so from that time, even before that time, studies were being done with vectors, vector-based vaccine delivery. And so this RV144 is a trial using a canarypox vector boosted with this GP120. It's in 16,000 people in Thailand. They had an interim analysis in 2007 and 2008. The trial is continuing, and we'll hear about the results of that study, I think, this year in October. And then there was the Merck studies, where they took an adenovirus-based vaccine, an Ad5 vector, I'll tell you more about that, and did something called the STEP study. That's where the title of my talk came from. Because the STEP study was a good idea. It was testing a very potent vector that can induce CD8 T cells that could have a chance of clearing virus early. And it was conducted first in North America and then in people who were adenovirus naive, Ad5 seronegative, and then extended to Ad5 immune people. And then it was extended to a sister trial in South Africa called Pambili. So these 6,000 people were studied with the Merck vaccine, and you see these big red X's. There's something that happened in this trial, and that was at the interim analysis when they were evaluating this first trial that was primarily in North America. They found that among the vaccinees, there was a higher rate of infection than among the placebo recipients. So the vaccine was causing an increased rate of infection, at least among some of the subgroups. And we'll talk about those subgroups later, but the consequence of that was that the Pambili trial was immediately stopped, and this PAVE 100 trial, which we had been planning for the last 7 years, which was slated to start on September 28th of 2007, 10 days after this Merck announcement, never started. So this PAVE 100 study that was going to evaluate a different gene-based vector concept did not start. So I'll tell you the basis for that vaccine product. I'll tell you the differences between it and the Merck product, and then I'll ask you what you think about starting the 505 study, and you can help me decide whether it should go forward or not. So the VRC vaccine candidate was multivalent. It included envelope genes from clades A, B, and C that covered about 85% of the incident strains in the global epidemic, attempting at least to have broader coverage of a greater number of viruses. And it included multiple genes, not only the GAG and PAL genes, but the NEF gene and these envelope genes. So it included a lot of genes and a lot of clades to cover a lot of viruses. And it used two different types of platforms, a DNA vaccine platform to prime the response, and an adenovirus-based platform to boost the response. The DNA was delivered by this needle-free injection system, and the boost had a set of matching genes. And so what we knew about that vaccine is that in non-human primates, among the vaccinated animals, you could lower the peak viremia relative to the controls, and you could lower the early set point viremia in animals who'd been vaccinated with a similar type of vaccine and challenged with SIV, simian immunodeficiency virus. We also knew that vaccinated animals had a survival advantage in this very stringent SIV challenge model, and so that DNA ad combination had better protection in monkeys than the adenovirus alone product from the Merck studies. We also know that in this example where compared to the sham or the mock vaccinated animals immunized just with the DNA ad expressing the GAG and PAL, which had a relatively small reduction in the set point viremia, had a much larger reduction in the semen viral load. So the amount of virus in the macaque semen that could be detected was even greater, more reduced in the vaccinees than you would have guessed from just looking at the plasma virus. We also know from early phase 1 clinical trials that giving either DNA alone or adenovirus alone compared to the combination of DNA and adenovirus, that the heterologous gene-based combination gave a much higher antibody response, more than 100-fold higher antibody response. And if you measure T-cell responses, and this is an ELISpot assay, but it's a measure of these specialized CD8 T-cells that I've been talking about, if you combine the DNA and adenovirus, you can see about a 5 to 7-fold higher response of T-cells in the combination than you can in either of the modalities by themselves. And the response was also qualitatively different. One of the big features of this new center is all the ways we can measure T-cell responses. And Mario Roeder and Rick Kaup there have pioneered this use of polyfunctional T-cell analysis using flow-based methods where we can phenotype cells and then define whether they're making these degranulation markers or putting out the things that can kill the target cell, or making these various cytokines and chemokines that are a measure of the CD8 T-cell function. And we can measure all these things at the same time on a single cell basis. So there's really a large number of phenotypes in these cells, but we condense them down into just how many functions are present. And we can ask, in the HIV response, is it polyfunctional or relatively oligofunctional? And what you can see in both CD4 and CD8 T cells, compared to just adenovirus by itself, the DNA ad combination is much more polyfunctional. There's many more cells that are making 3, 4, or 5 functions instead of 1 or 2. And so in the DNA ad heterologous prime boost combination, we're seeing more T-cell response and a more functional T-cell response. So this product was moved into phase 2 studies, and this was done on 3 continents and 3 major regions. by three major networks. And so we had network partners, the HVTN, which was domestic and also in South Africa, the U.S. Military Research Program, and the International AIDS Vaccine Initiative, a nonprofit and a military program, worked together in East Africa with a very diverse group of investigators, and we accomplished a 920-person study we called the TRIAD. And in that, I'll just show you this one table. And the bottom line is that either at our facility in Gaithersburg, the HVTN labs in Seattle, the IAVI labs in London, or the military field site in Tanzania, if you measure T-cell responses in the field in these vaccines in populations at risk, you could see about a 70 to 80% response rate, which was considered good at the time. But then on September 18th, ours is September 18th. It was even worse than September 11th in some way. But this report from the Merck study showed that there was increased rate of infection among vaccinees compared to placebos, and it was even more profound in people who were previously exposed to adenovirus 5. So in AD5 seropositive people, there was a odds ratio of someplace around 3 to 1 vaccinees to placebos getting infected. So the question is, first of all, what's going on there? And second of all, is it safe to do any more gene-based vaccine studies? So the next analysis broke it down into whether a person's AD5 seronegative or positive, and whether they're circumcised or uncircumcised. And what you see here is the greatest risk among vaccinees was among the uncircumcised men. And this entire analysis was done in men. So of the 83 infections in that first analysis, 82 of those infections were in men. So this analysis is only evaluating gay men in North America. And so the odds ratio is highest in uncircumcised, whether they're AD5 seropositive or not. But there also appeared to be some risk among circumcised men if they were AD5 seropositive. And so the question is, if you just focus in on this group of circumcised AD5 naive individuals where the relative risk of vaccination for infection was less than 1, could you do a safe vaccine study in that population? The other piece of data that came out of the Merck study was that among people who had a low AD5 antibody before vaccination, if you looked at their ELISpot response compared to the viral load after they were infected, there appeared to be a correlation in those people who are AD5 naive and AD5 seronegative, as opposed to the people who were AD5 seropositive prior to vaccination, where there was no correlation at all. And so the question became, was it safe to do a vaccine trial in AD5 seronegative circumcised men and ask the question, can we identify a T-cell measurement that correlates with reduced viral load and at least take another step toward finding an HIV vaccine? So that's one piece of information that you need. Now I'm going to compare the VRC vaccine to the Merck vaccine, and you can then judge if it's different enough. So the VRC vaccine includes these plasmid primes with an adenovirus boost. It's an AD5-based vector. It has some differences, but the GAG-PAL included in ours is represented here. In the Merck vector, there was a GAG-PAL NEF adenovirus given repeatedly at 0, 1, and 6 months. So the genes differed somewhat. Our product included GAG and PAL and NEF, as did the Merck product, but we also included envelope genes, and so there was a little bit difference in antigenic content. And then this is a nuance to some people, but it may be one of the most important differences in these two vectors. There's different ways of making adenovirus vectors. The Merck vector used the E1 region to put the transgene, so the GAG and POL and NEF were put here, so that when the adenovirus was used to deliver the gene, those genes were expressed out of this site. But that means the rest of the genome backbone for the adenovirus was also making mRNA and making adenovirus proteins. In our construct that was made by Genvec, not only was the E1 region used for expressing the transgene, but the E3 region was deleted, partially deleted. That's a region that adenovirus uses to modify immune responses. It has analogs to different chemokine receptor genes. It has proteins that will downregulate the MHC molecules. And so this contains a lot of immune avoidance genes. That was deleted. And importantly, this E4 region was deleted. And the E4 region has a set of genes that allows the other adenovirus genes to be expressed. And so if you just look at the Western blots here of the kinds of structural proteins in adenovirus that are made by either an E1 deleted or an E1, E3, E4 deleted vector, what you see is the hexon and the penton base and the fiber that makes up the capsid of the adenovirus are made from the E1 deleted vector, but not made. So there may be a lot more adenovirus proteins being made if you don't delete the E4 region. So that's the other difference in these vector systems. So in the field studies, we saw that not only in the phase 1 studies, but in the field studies, 6 months after the adenovirus boost, you were able to maintain a fairly high level of T cell response to the different envelope A, B, and C proteins or peptide pools and the GAG, not so much against POL or NEF. But against the envelope and GAG proteins, even after 6 months, there was a good T cell response. So the question is, and this is what we believe, and I'd like to hear comments from the audience about whether I've made the case to you. That is that we think it's safe to proceed with this type of product in Ad5 antibody-negative circumcised MSM. Because of the partial protection in SIV-challenged monkeys, we think we have a slightly different possibility for efficacy. Because there's differences in the antigenic content and the pattern of immune responses with a more balanced CD4, CD8, and more envelope in GAG rather than POL and GAG response. And because, importantly, we have the opportunity to identify T cell responses that could correlate with reducing virus load, which we'll need to take the next step in vaccine development, we think that there's a good scientific basis and ethical basis for moving forward. So there is a trial called HVTN 505. It's much smaller than that original PAVE 100 study. It's designed to study this vaccine in this group that I just described, and it would enroll about 1,350 people, half of whom would receive this vaccine and half of whom would not. And the major endpoint would be to see if they could reduce viral load by at least a log in those people who became infected despite being vaccinated. So if we were able to start that study, and it has now been submitted to the FDA as of just a few days ago, accrual would begin around July, and that means that sometime in 2012 we would know if this vaccine was having the effect that we thought it might have. So I'm going to stop there for just a moment, and I'd like to take a few questions, and then I want to go on to finish what alternative strategies there may be. So if anybody has a question or would like to make a comment here, let me hear it.
The Merck study was done just in men, is that correct? And if so, why?
Well, the Merck study was done initially in people who are Ad5 seronegative, and then in 1,500, then it was extended to Ad5 seropositives in North America and the Caribbean. It turns out that about 1,200 of those 3,000 were women, 1,800 were men, but almost all the infections were in men. It says a couple of things. One, it says that we aren't very good at identifying high-risk cohorts of women in the United States. It also probably says that women follow directions better than men. So you can interpret it either way.
Barney, in terms of phase 2s for something like this, how much does putting on the trial change behavior and things?
Well, when you go in to start preparing sites for a vaccine trial, the question is, how much does preparation change the dynamic of where you could do the trial? So when you start preparing sites like this for vaccine work, it often takes years of preparation of cohort development, education, testing, and treatment exercises. And so generally, if you go into a place with a serial incidence of 3 to 4% a year, after a year or two, that incidence will be halved. It will go down to 1 to 1.5% per year. So in general, the incidence will go down about 50%. It's hard to get it to go below 50% with just educational intervention, but it does start making a big difference in the number of infections you might expect from a given cohort.
Barney didn't make it completely clear in my mind. Are you testing this on them or us, and that you define the them or us? And I'm pleased that you're not filling off any more primates at this time. I have strong feelings on that. And what's your deadline going to be? How many failures do you figure, okay, no more?
So the question is where we're going to do the trial.
Yeah,
I think the trial as it's planned right now, originally it was going to be planned in those same regions where we did the phase 2. But because the seral prevalence of AD5 is so high, especially in Eastern Africa, it's about 95%, recruiting a population of AD5 seronegative people there was untenable. And because of the issues involving safety, the trial is going to be done in North America, where at least the AD seroprevalence rate is in the range of 50% instead of 95%. So we at least have a chance of enrolling people. So this trial would be planned for North American MSM, men having sex with men, AD5 seronegative, circumcised men. The subgroup in the Merck study that actually may have had a benefit from the vaccine at least had a lower odds ratio for infection with the vaccine. The second question is how many times will we fail with an HIV vaccine approach before we stop testing and developing HIV vaccines? Well, if I tell you that there's 7,000 new infections every day with HIV, do you think we should stop trying? Okay, so our goal is to never stop trying, and either we're going to find a vaccine or we're going to change the dynamic of HIV in the world by trying to develop a vaccine. And the question is, what is it going to take to develop a vaccine? And I have I'm going to give you a few additional slides about some of the new approaches that are being taken and some of the technology that's being applied to this problem, and hopefully to get us to the point that we can have a more effective AIDS vaccine. But, if you look at the malaria field, there's been at least 20 to 30 failed efficacy trials for malaria vaccines. If you look at the development process of almost any other vaccine product, you have to go through a lot of failure to ever achieve something that's working. We're still working to try to improve the influenza vaccine, something that's already licensed. So I think that in vaccine development in general, there is a lot of failure before success, typically. So one of the problems for achieving the kind of breadth we need, not only the breadth of response in an individual, but the breadth of response in a population to cover all the possible incoming HIV viruses, is being addressed in a bioinformatics approach. And Betty Korber at Los Alamos is a brilliant mathematician who also is a trained immunologist, and she has brought a lot of innovation to HIV vaccine concepts. And so one of the things that she's done is using bioinformatics, she takes sequences from a large population of individuals. So for instance, all these different lines might represent a population. Well, these would be the different viruses within a population. And then she puts it through her computer algorithm and asks, if I start recombining these sequences at natural recombination break sites, because HIV does a lot of recombination, and I keep remaking recombinations of viruses, and then I ask, within those new virus recombinants, how many of the T-cell epitopes that have been mapped within these viruses have been retained? And can I achieve a virus sequence that's relatively natural with the normal breakpoints, but that has achieved a higher level of epitope coverage for the different virus possibilities? And what she's found is that by doing that, she can find winning sequences from each of these various recombination experiments, and then by using a combination of these in silico recombined sequences, and she can achieve about a 20 to 30% higher level of coverage with new epitopes above any given single natural isolate, or even a combination of natural isolates. And that's now proving to be true in animal experiments. So if you use a cocktail of 2 or 3 mosaics, or even 4 mosaics, compared to a cocktail of 3 natural isolate sequences, you can achieve a much broader range of response. And so instead of having the 3 or 4 epitope response in a normal setting, you might achieve, in some of the monkey studies, they've gotten up to 12 to 20 epitope responses. And so the thought is, especially in T cells, if you can achieve multiple responses to multiple epitopes, it would be much harder for that virus to escape. Just like 3 drugs is when we really started making a big impact on HIV, instead of just single treatment where the virus could serially escape. So this new concept of mosaic inserts is now being combined with a whole variety of new vector choices that are being developed, not just DNA and Ad5, but there's a whole variety of new gene-based vectors that are being used and combined in different ways, using these new insert designs to see if we can really maximize the breadth and the magnitude of the T-cell response. But the real problem is antibody, and so I'm going to end with a little brief series of slides on the antibody problem. And most vaccines have worked because we can introduce an antibody, and it turns out for various reasons, because this protein, this GP160 protein on the surface of HIV is so highly glycosylated, it's hard for antibodies to reach the most vulnerable sites. It's very flexible, and so there's a lot of different forms that it takes before it locks into the receptor, making it hard for antibody to ever lock in on a single conformation. And the sites that are the most vulnerable are hidden in these places. If you were looking at the top of this molecule, really in a groove that requires an antibody coming at just the right angle, of just the right specificity to neutralize. And so over all these years, there's only been a handful of antibodies, monoclonal antibodies, that have been found to broadly neutralize the commonly transmitted strains of virus. 2 of them are against this membrane proximal region in the GP41 that is anchoring the protein to the viral membrane. And then 2 of them are in this GP120 head. One of them is where the CD4 binding site is for the main receptor of the virus. That's the B12 antibody. The other one is 2G12, which recognizes a combination of glycosylation sites. It's not particularly well understood. So the question is, if you apply structural biology to understanding exactly the atomic structure of these GP120 in their different forms, can you use structure-based vaccine design to find something that might actually work for neutralizing the antibodies? And so one of the big findings early on using this structure-based approach is that if you compare this core structure over here, which Peter Kwong solved in 1998, this is taking off a lot of the GP120, just looking at the core molecule and asking in the CD4 bound conformation, bound to its receptor, what does that GP120 look like? And this domain right here, which is called the bridging sheet, this is where the CD4 binds. This is the inner domain and the outer domain. This is the place that binds the co-receptor for HIV, the CCR5 or CXCR4. And so this was one of the early important vaccine targets, thinking that we could really use that as a target for vaccine development. Well, it turns out a few years later when Harrison solved the unbound, unliganded structure, if you look at GP120 before it's bound to its receptor, these two regions that make up the bridging sheet are not even together. And so that region actually doesn't exist in the virus before it's already bound onto its receptor. So using this kind of structure information, we can have a much better idea of what the targets are for HIV. And so making antibodies to the bridging sheet is obviously not an option based on this. And in contrast, if you focus not on the inner domain and its changing position, but the outer domain in both the unliganded and liganded position is very constant. So this region of the GP120 molecule remains very constant. And so Peter and his group at the VRC have used a series of experiments where they co-crystallize these antibodies, these broadly neutralizing antibodies, to the GP120 or the GP41, and they determine exactly what the structure of the binding site is for neutralization. And one of the more important papers was this one, where they used the B12 antibody pictured here, interacting with the CD4 binding site here, and then they mapped the region of the antibody footprint to the CD4 footprint to try to really show exactly how the antibody was binding to neutralize the virus. And so if you have that information, then what can you do with it? So the different approaches now that are being taken with the proteins based on this structural information is the following. First of all, one of the problems with HIV is that this region right here where CD4 binds is somewhat blocked by the other trimers, and these regions in here, if you just present the single monomer, if you just present one of these to the immune system, all the antibody response is happening to this, which is not even present in the natural virus. And so one option is to make the more native trimer as a vaccine, hoping that the antibody will find that little groove right there to go into. The other approach is to make an artificial protein in which you try to single out that one area where you want the antibody to bind by cloaking the rest of the protein in glycosylation sites. So you can build these glycosylation sites into your protein so that this now is covered with sugar, and you're hoping that it'll make the antibody focus on that one green spot. Another approach is just simply presenting the outer domain of the protein to the immune system, hoping that it will see that CD4 binding site. An even more elegant approach is scaffolding, where that exact structure for neutralization is preserved. And a guy at Washington University of Washington in Seattle, Bill Schief, takes that structure and he does a threading program. And so he threads that structure through the entire protein database, structural database. And he finds other proteins that can hold that structure in the right conformation. And so you can then lift that exact determinant and put it in 10 different proteins that are irrelevant in and of themselves, other than the fact that they can hold that one little structure in just the right shape. And so all these approaches are being taken, and some of the more successful approaches have been the trimer, in which some of the pieces have been locked into place with disulfide bonding so that it won't be such a flexible molecule and will have less entropy. That approach is starting to show some promise. And this scaffolding approach and the outer domain approach is also starting to show some promise that we can start directing more of those antibodies at the right angle to the right spot. But the question is still whether we can ever develop that into a real manufacturable vaccine, which is still several years away. So that is what's happening in HIV vaccine research. We still have a long way to go, but we still believe that someplace over here, after we finish walking on this path, that we will get there eventually. And I'll stop there and take any more questions. Thank you.